inflammation, and complement cascade. The innate immune system is the first line of defense, which includes physical barriers such as skin, chemicals in the blood, and immune system cells that attack foreign cells in the body. Bacteria enter through skin only when skin is damaged. And if the pathogen successfully infiltrates to our body, we have complement protein in our blood vessels that enhance the ability of antibodies and phagocytic cells to clear pathogens from an organism. The complement protein do many things. First, opsonization. This is the process where the complement proteins coats the outer surface of pathogen, which allows phagocytes or macrophage to engulf the pathogens more easier. Next, membrane attack complex, which called it MAC, is a structure typically formed on the surface of pathogenic bacterial cells as a result of the activation of the host alternative pathway, classical and lactin of the complement system, and it is one of the effective protein of the complement which causes lysis and destroy the pathogens. Third, enhance the inflammation. The complement system composed of more than 30 proteins. These complement produced in the liver, then they travel through the bloodstream, and these complementary proteins circulating in an active form, and so they don't attack the body. But when they cross by pathogen and they become activated, they opsonize pathogens and create MAC on pathogen and also enhance inflammation, and this happens when the complementary protein interact with each other and induce cascade of activation in 3 different pathways. The first one, classical pathway, which involve C1, QRS, C4, C2. Alternative, which involving factor B and D and C3. Lactin, which involve mannose binding lactin, C4, C2 mannose. And this is surplus C3 and stimulate membrane attack complex to activate mast cells, lysis, or opsonization. We'll start first with classical pathway. When the pathogen bind to antibodies, it will activate C1, QRS, C4, C2 to bind to bacteria and produce antigen antibody complex, and this is from complement protein complex. This is explain the pathogens and the antibody, and this is the complement protein complex, and after the binding enhance the phagocytosis and produce C4b2A, or called C3 convertase, and this is surplus C3 to C3A, which enhance inflammation, and C3b, which activate opsonization and cell lysis, MAC. This is the opsonization molecule. It's coated to the surface of microbes, and hence opsonization. The phagocytes has a special receptors called C3b receptor to enhance the phagocytes and produce membrane attack complex. This is before the cytolysis, and this is after the cytolysis. The classical pathway, when the microbe binds to the antibodies, it will activate the C1 to C1 split C2 to C2A and C2B and C4 to C4A and C4B, and this is the combine. It's split C3 to CA3A, which enhance inflammation, and C3B, which activated cytolysis and opsonization. Alternative pathway, no antibodies involved, only the microbes. It's bind to the different factor B and D and B, and this is split C3 to C3A and C3B, which activated cytolysis and opsonization, and produce C3BBB, the C3 convertase, activated split C3 to C3A, which enhance other complement and produce C3A and C5A, which stimulated mast cells, and C3B, which activated opsonization and produce attack membrane complex. And mast cells, it's after binding to C3A and C5A, it's produce activated secrets and histamine. Now the lectin pathway, it's produced by the liver, and this band carbohydrates called mannose binding lectin, and this is in the bacterial cell wall and some viruses, and this is activated C2 and C4 and split to C2A, C2B and C4 to C4B and C4A, and this is the combine. It's split C3 to C3A, which enhance inflammation, and C3B, which activated cytolysis and opsonization. Thank you.